Welcome to Miss Esco's Exploring Inheritance. We are going to start exploring inheritance by first looking at Gregory Mendel. Gregory Mendel is coined as the father of genetics. He was an Austrian monk that coined the idea of studying heredity. He looked at a variety of different experiments that all looked at the basic patterns of inheritance, and then from those patterns of inheritance he actually looked at how these traits were going to be inherited. Gregory Mendel was up against the blending hypothesis. The blending hypothesis was the founding early idea of genetics, and it was the idea that everything that was put together in our genetics was a blend. So you would put a red parent flower and a yellow parent flower together and you would get orange. It was easy to see that humans, animals, were a blend of the mother and the father. Over a bunch of different generations and hypotheses that Mendel put together, this blending hypothesis was disproved. We will be coming back to this idea of a blending hypothesis because we do have some genes that will interact in a way that looks like it is a blend. Gregory Mendel's experiments started with his plant breeding experiments. He looked at the characters and traits within pea plants. Why pea plants, you ask? Well, they grow quickly, and they had 8 different characteristics that we could look at. So you see over here, you've got seed, and then within that seed you have the wrinkled or the round, and then is it yellow or is it green? The flower color, is it white, is it violet? The pod itself, was it full or constricted? Was the color of the pod yellow or green? And then just the stems, were they short, were they tall? These characteristics gave Mendel lots of different things to look at. And when we start to talk about characteristic and trait, the character is the larger, the trait is the smaller of the two. So you have flower color, and then you have the white or the violet. You have seed form, round or wrinkled. From these, he started his entire experiment. He started by producing pure plants. Pure plants are those that were self-pollinated. So here's this short plant. I'm going to pollinate it, and plant those seeds to get all short plants. This became known as the parental generation. Your parental generation is the pure trait. So in this little picture, you have the pure trait for purple, the pure trait for white. Then from there, the parental generation or your pure generation, Mendel then cross-pollinated. So he would take this small or the short plant, crossing it with the tall plant, or in this picture, taking and crossing the purple pure plants with the pure white plants. This would then produce your F1 population. In our picture, the F1 population is all purple, and we'll kind of come back to why did we get all purple and no white, but this is where Mendel kind of started with. So he starts with seeing pure plants, getting all pure plants. Then he crosses and questions, why am I now getting only purple plants? Then took those purple plants and self-pollinated, and all of a sudden here comes back in the F2 generation those white plants again. Okay? So this is kind of the process that he went through, was looking at all of these different generations, and he spent 7 years just pollinating plants and cross-pollinating and then self-pollinating again to produce those F1 or F2 plants to see what he found out. So from Mendel's experiments, he learned 3 different large principles. He has his F1 and the F2. And from those, he got the idea of dominance and recessive, so that the dominant is going to mask the recessive, and then the recessive will come back when it is not masked by the dominant. So in the picture that just popped up, you're going to see that here is the difference between the lobed earlobe or the attached earlobe. The little e is the recessive alleles, both showing that they have the attached earlobe. All of the other ones have an actual lobe because you've got that big E, or the dominant factor, that is going to be seen if it is present. Mendel's experiments also led to 2 other principles: segregation and independent assortment. Segregation is the idea that the alleles that you get from parents are going to be segregated. So you're going to get one from mom, one from dad. Two factors that are separated. The offspring will get one from mom, one from dad, so that every single time that those are formed in the gamete, they're going to be different because of all of those things that we talked about with meiosis. So each one of those gametes is specific to that gamete, which then when those two gametes come together, you're going to have a segregation of factors. The third principle was the independent assortment. Independent assortment is that factors for differing characteristics are not connected. So just because you have blonde hair does not automatically going to mean that you have blue eyes. They are not connected. So just because they happen to be on the same allele, we know that they are not always passed together. This is that idea of that crossing over and bringing in more genetic variation that we talked about with meiosis. So where does this leave us now? This leads us now to taking Mendel's 3 principles and putting them into action in our genes and our chromosomes and our alleles, and actually being able to use those 3 principles to predict what we're going to see. So, gene is a segment of DNA, okay? That should be a review for us. But within each one of those genes, we're going to have alleles. And alleles are alternate forms of the gene that code for the trait. Remember, the trait is the specific thing. So if we're talking about flower color, it's going to be the colors. So the purple or the white. Or as the example on the screen says, tall or short. Those are the traits. The characteristic would be height or flower color. We represent our alleles with letters. So you're either going to have a capital letter for a dominant or a lowercase letter for a recessive. Okay? So here on the picture that just popped up, you have your genes. Okay? There's the allele for the purple flower. There's the allele for the white flower on the homologous chromosomes. So then that moves us into being able to actually start to look at how are we going to use this process. Couple more terms that we have to get underneath our belt. Genotype and phenotype. Genotype is the actual genetic makeup. Phenotype is the external appearance. Okay, so you have your genes that code for what we're going to look like. Okay, when we see each other, we see our phenotypes. We do not see our genotypes. Then we move into when we start to put these letters together, either capital or lowercase, how are we going to talk about our alleles? We talk about our alleles as either homozygous or heterozygous. With homozygous, you can be homozygous dominant for a gene or homozygous recessive for that gene. Meaning that dominant, I have 2 alleles that are both dominant. Recessive, 2 alleles that are both recessive. Okay? Heterozygous, 2 different alleles for that gene. A dominant and a recessive. Right. In this picture, it's kind of showing, again, there is the allele on the gene. Okay, you have a big B, that's going to be your homozygous dominant. Big B, heterozygous. Little b, big B, heterozygous. Homozygous recessive, then little b. You're going to get annoyed with how many times that we say little b, big B, capital B, little b. Okay, it's just kind of the process of how we work through our genetics. The next step to preparing for all of this prediction is actually talking about probability. And probability is the likelihood that a specific event is going to occur. We can record these things in a variety of different fashions. Decimal, percent, fraction. So when we start to go over our actual problems, you will hear me say and write them in a variety of ways. I want you to pick the one that you are the most comfortable with. If you like fractions, use fractions. If you like percents, use the percents. How we calculate the probability is the number of one event over all of the events. So when we start to talk about using them in our Punnett squares, if we have 4 boxes or 4 probability, that would be our total number of events, and then each one that we actually see would be each kind of event. So we will be talking a lot about probability because we're going to be using these Punnett squares to actually predict what the outcomes of a genetic cross will be. So our first one is going to be crosses of a monohybrid cross. Mono meaning 1. Okay, so we're talking about one trait or one characteristic with 2 contrasting traits. All right, so we're going to be using the example of T, big T's and little t's. The t's are going to represent tall or short. So if you see little t, that's going to be short. Big T, anytime you see that, that is going to be your tall. Okay, so in our first trait, here's our Punnett square that we have all set up for us. We've got 4 boxes, and we also have 4 alleles. So when we start to organize them, we're going to take and we're going to put our alleles on the top of the boxes. Okay. One question that you should be having right now is, does it matter if I had put the little t's on top or the little t's on the side? Does not matter. Okay, as long as I keep them together. I wouldn't have wanted to take a big T from here and a little t from here and put them together. Okay, then it's as simple as filling down and filling across. So it's going to be the big T, little t, big T, little t. We do want to make sure that when we are filling in that we're putting our capital letter first. Okay, it's going to be a little bit easier to read when we go to figure our genotypic and phenotypic ratios. Okay, so genotypic again, genotypic means what we actually see. So if I'm looking at all of these for their genes, every single one is the same. So I would have 4 over 4, which is equal to 1 because I want to reduce. Or I could also say I have 100%, and yes, you should be making fun of my writing right now, 100% big T, little t, or I could also call that heterozygous. Okay, so I can either write out the letters or I can write out the actual word, heterozygous. Then I go to the phenotypic ratio. Because I have all of the same genotypic ratios, I'm also going to have 100% probability of tall plants. So every time that I cross a tall big T with a short little t, I will get a tall plant. All right, so let's keep going. So now we're going to cross a homozygous dominant, big T, with a heterozygous, big T, little t. So again, does not matter where I put my alleles as long as I keep them together. So here's my heterozygous dominant, or my homozygous dominant, and here's my heterozygous. Again, I'm going to fill across, and I'm going to fill down. Okay, this time we should be seeing that we've got different things inside. So now I've got 1, 2, or 2 over 4, which reduces to 1/2, which is also 50%, or 0.5, because remember we can also talk about them as a decimal. Okay, is big T, or homozygous dominant. Okay, then I look at the rest of my Punnett square. The rest of my Punnett square, again, I have 2 over 4, which reduces to 1/2, which is also 50%, or 0.5, big T, little t, or heterozygous. Okay, now for my phenotypic ratio, I've got to kind of stop and look at this. This plant, big T, is going to be tall. Big T, tall. Okay, then again, here I've got big T, little t. The minute I see that dominant allele, I know that that plant is going to end up being tall. So I have a 100% chance of having a tall plant from crossing the homozygous dominant and a heterozygous. So again, I've got 2 different things that I'm starting with, but my phenotypic ratio is the same as the problem before. Okay, going back to Mendel's idea that the dominant allele is going to always mask that recessive. Third problem. Now we're going to cross a heterozygote and a heterozygote. Okay, so again, pick your sides. Fill across, fill down. You're always going to want to show me work. Okay, every problem that we do, you're going to want to show me work, because if I can do this part right, but then I maybe get a little bit messed up when I'm doing my ratios, I'm going to still give you points over here. Okay, so you always want to make sure that you show your work, even if you think that you know what everything is going to look like. This time we're going to use some fractions, okay, and we're just going to count how many times we see big T, big T. I see big T once. So I'm going to have 1 over 4 big T, or homozygous dominant. My next option is big T, little t. How many times do I see big T, little t? Twice. So that would be that 1/2 big T, little t, or heterozygote. Then I've still got one left. I have one left of the 1/4 chance of little t, little t. Okay, now my phenotypic ratio. My phenotypic ratio that I'm going to look at is this one's going to be tall, these two are going to be tall. So I have a 3/4 tall and a 1/4 chance of being short, of seeing a short plant. So now, like Mendel predicted, when I actually cross a heterozygote with a heterozygote, my recessive phenotypic ratio can come back out. So in this case, when we were talking about the purple and white flowers from earlier, this is that white flower coming back. This is that short plant coming back. And this 3-to-1 ratio, when you cross a heterozygote with a heterozygote, was Mendel's bread and butter. This is where he started with everything. And when we know that we cross a heterozygote with a heterozygote, we will always see a 3-to-1 ratio in the F2. Okay, now this is the predicted. When we talk about predicted, it's not always going to match the actual. We would have to have thousands and thousands and thousands of pea plants to actually get the predicted. But we can still use this as a way to give ourselves an option of, will this material show back up? All right, next idea is the test cross. Okay, test cross is going to be ridiculously confusing. Okay, but this is a test cross. It is something that is done on paper. We are not talking about something that is physically done. Okay, so long story. I'm gonna have guinea pigs in my basement. Okay, guinea pigs in my basement. I want the elusive white guinea pig, but I have a black guinea pig. So before I start breeding guinea pigs and end up with hundreds of guinea pigs in my basement, I want to know what is my actual probability of getting that white guinea pig. So I take my black guinea pig, which I don't know if it is, I know it's dominant, I know it has the dominant phenotype, okay, but I have no idea what its genotype is, okay. I'm not a teacher, I don't know any of this stuff, but I really want to get into the guinea pig breeding business. So I know that it's either got to be a homozygous dominant or a heterozygote. Okay, so that's where I get into big B or big B little b. Okay, to then test what my probabilities are, I'm going to cross both of my possible genotypes with the recessive, that elusive white guinea pig. I'm not really doing this. I'm sitting at my kitchen table giving myself some data on if I want to actually start breeding guinea pigs. So when I fill these, I see that every single one is going to be the heterozygote. Okay. Now, when I go to this side, I get a little bit something different. All right. If my guinea pig actually happened to be a heterozygote, I seem to have a 50% chance of getting that elusive white guinea pig. All right. So by looking at this, if I have a homozygous dominant guinea pig, I have no chance of getting the elusive white guinea pig. I just got the generic black one that I'm going to have to sell at a very cheap rate. Okay. But if I happen to have the heterozygote of my guinea pig genotype, then I do have a 50% chance of possibly getting a white guinea pig. So then, because of these results of the test cross, I have to make up my mind on saying, do I actually want to start breeding guinea pigs in my basement? Okay, that's what the test cross is for. It gives you information, and then it allows you more information so you have an informed decision. Next thing, the dihybrid. Okay, so if mono meant 1, di is going to mean 2. Okay, so now we're talking about 2 characteristics and 4 contrasting traits. All right, so tall and purple, short and white. The first step is that we have to figure out all of our gamete possibilities. Our gamete possibilities are going to come from that lovely math thing that we call foiling. So we're just going to take all of one allele and distribute it to all of the other. And then we write it all out. So I get big D, or big B, big D. Big B, big D, little b, big D, little b, big D. Now,
Here you should notice that I left the little letter in front. That's going to become very important when we actually put it together in this huge Punnett square. Okay, we want to make sure that we've left the little one first so that it's easier to put back together with the B. Alright, so then we do our second one. Just FOIL everything out, see what we get. Okay, we're going to have big B, big D, big B, little d, little b, that was a bad B, sorry, little d, and then little b, big D. Okay, and I reversed those two, I apologize. But you're getting the idea. These are all of our possible gametes from these alleles, all of the possible gametes that could be produced by these combination of genes. So now we would take this, and now we're going to put it into a 16-box Punnett square. Okay, so again, I'm going to have to FOIL everything out, and we're going to see that in this example, at least, we end up with all of the same gamete combinations. So for every one of the first one that's homozygous dominant and homozygous dominant, we have a big T and a big P, tall and purple, short and white. Okay, for the next one, because they're all homozygous recessive, we see that we're going to get all little t, little p. Then it's just like we did with the 4-box Punnett square, except now we've got more. And we're going to fill across and we're going to fill down. So while we're filling across and filling down, though, we're going to put the t's and the p's back together. Okay, always putting the capital first, then the lowercase, until we see the whole Punnett square is full. Big T, little t. Okay, I hope that we're kind of seeing that because all of our gametes are the same, that all of our probabilities are also going to be the same. Okay, so then when we start to look at our genotypic ratio, we've got a 100% chance of having big T, big P, little p. Okay, or homozygous or heterozygous for both of the alleles. Then we also have a 100% chance of tall, wow, that was bad, tall and purple. Trying the cursive with my mouse here. Alright, so this is the probability if I cross two homozygous dominant with two homozygous recessive. I'm going to have 100% heterozygote for both of my genes, and I'm going to see both of the dominant traits. I'm going to have the tall and the purple. Okay? Now, the next one is going to be a little bit different, as we're going to have different alleles for all of them.
Alright, so now on to a little bit harder of a dihybrid cross. This one we're going to be crossing two heterozygotes with another set of two heterozygotes. Okay, this one we still have to start with figuring out all of our gametes. So that's where we're going to use that FOIL method and take all of each one of the gametes to each other. Okay, because they are the same, they are going to be the same on the top and on the bottom. So we would end up with a big T, big P, big T, little t, big P, little t, little p. Okay, same thing down the other side because we have the same. Okay, if we have an example or a problem that is not the same, you wouldn't have the exact same on both sides. But because we're crossing two heterozygotes, we will have the same. You'll also see that this one will be very unlike the other one where everything was the same inside the boxes. This one is going to be very different. Okay, so we're still going to fill across and we're going to fill down, keeping our T's and our P's together. So big T, big T, big P, big P. Okay, so I took that one and that one, put it together. Okay, so I'm going to work down first. So now I have another big T, another big T. So I got two big T's. Then I've got a big P from up top, little p from my side. Now I'm going to take big T, big P, and put it with little t, big P. Okay, always keeping my T's in front, my capitals in front of my lowercase. Okay, and then I continue. Big T from the top, little t from the side, big P from the top, little p from the side. Now I'm going to move over to my next row. Big T, big P, little p. Big T, little p, little p. Big T, little p, big P, little p. Big T, little p, or little t, little p, little p. Okay, now next row. Big T, little t, big P, big P. Big T, little t, big P, little p. Big T, little t, big P, big P. Little t, big P, little p. Okay, and our last four. Big T, big P, little p. Big T, little p, little p. Little t, big P, little p. And then finally, little t, little p, little p. Okay? When you're going through here, please make sure that you're making a difference between your uppercases and your lowercase letters. Okay? I have to be able to tell from your handwriting, and yes, I know that mine is pretty bad. Okay? I'm using a mouse. All right? But you can still tell which ones are uppercase and which ones are lowercase. Okay? Also on my T's, I was making that little tail kind of thing on my lowercase so that they did look different from my uppercase T's. Okay? Now, after this, the next step is figuring out our genotypic and phenotypic ratios, just like we've done for all of the other problems that we've looked at or examples that we've looked at. Okay? This one, however, we've got to make sure that everything adds up to 16. Okay? The easiest way to kind of go about this is going to be to start with your homozygous dominant. Okay? So we're looking for how many times do we see big T, big P, big P. Okay? Scan through all of them. You should see that we only see it once. So that would be 1/16 big T, big P, big P. Okay? You're going to want to probably use the fractions for this one just because it's easier to add up all of your fractions to make sure that you've gotten all 16. Okay? Also, you're not going to want to write homozygous dominant. You're going to want to write the letters. Okay? Just kind of saving yourself a few little steps. The next combination that we're going to go for is going to be big T, big P, little p. Okay? So kind of sticking with a trend. Then we'll look for big T, little p. Okay, so that we're getting through and making sure that we're looking for every single option possible. So my next one would be two big T's, the big P, and the little p. Okay, I see it once, scanning, and I see it again. Okay, so this time it would be 2/16, or we would reduce that to 1/8. My last one in my homozygous dominant T's, okay, I'm going to see that we only have one, okay? Kind of giving a little different things so that I'm marking them as I go through. Again, 1/16. Then my next step is going to be to look at all of the combinations where I could have a heterozygous T, okay? So heterozygous T, then my homozygous dominant P's, my heterozygous P's, and then my homozygous recessive P's. Okay? This one, you're going to see that if I see a heterozygote, I've got 4 here, and then I've got them replicated over here. So they're like mirror images of each other. So if I'm looking for my big T, big P, I see it here, and I see it here. Okay? So again, I see the 2/16 or 1/8. Okay? My next one would be looking for both genes being heterozygotes. Okay? I see it here, I see it here, I see it here, and then I see it here again. Okay? So now I've got 4/16. Okay? You should be asking yourself, What does that reduce to? Okay, and that's 1/4. Then my last one would be the heterozygous and the homozygous recessive. Okay, I see it here, and then I see it here again. Okay, so then we've got the 2/16. And yes, I'm writing out every single fraction because at the end, I'm going to come back and I want to be able to count and make sure that I have all 16. My last genotypic ratios that I'm going to look at are going to all have the homozygous recessive T's. Okay, big P with this one, big P, and then little p. Okay, in this example, we have every single one of our options. Okay, this is not always going to happen. Not every single problem is going to have every combination that the genes can have. You go off of what is in your Punnett squares. Okay, so now we're finally down into just the last 4, and I see my 1, little t, big P. So I've got my 1/16, then I've got 2, one here, of my little t, big P, little p. So I've got the 1/8 or the 2/16. And then finally, my last but not least, 1/16 of my heterozygote. Okay? So now to just make sure that I have, and I've matched all 16, I go back and count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So now I know that I've got every single genotypic ratio possible. Okay? Now the phenotypic ratios. For the phenotypic ratios in the dihybrid, we're talking about 2 traits. So we're going to have the options of being tall and purple. Okay? Or the plant could be tall and have white flowers. Right? Okay? And what's my other option? It could be short and have purple flowers. Okay? Or short and show the white flowers. So now I've got to count up all of my different combinations of these. Again, why it's going to be so helpful to have my whole numbers out there so that I don't miss anything. Okay, so I'm going to start with the tall and purple. So for tall and purple, I'm going to be looking for a big T and a big P in any combination because those are my dominant alleles, and the dominant allele will mask the recessive. So I see it here, I see it here, don't see it here, here, and here. Okay, those are my options. So now I'm going to count. 1, 2, 3, 4, 5, 6, 7, 8, 9. So I have 9/16, which cannot be reduced, chances of having a tall and purple plant from this heterozygote cross. Okay? The next one would be tall and white. So for tall and white, I'm going to be looking for the dominant T and then the 2 recessive P's. So I've got it here, and then I've got it here again. Okay? Those are my only options because over here, I've only got little t's. So I have 1, 2, 3. So 3/16 tall and white. Then it only leaves me these options over here to be either short or purple and short and white. Okay? So let's start with the short and purple. Short and purple will have the 2 little t's and 1 big P. Okay? So I see it here and I see it here. Okay? So 1, 2, 3. 3/16 again. And then finally my short and white. The only ones that will be short and white will be my last little chunk here, and that's 1/16. If you remember back to our monohybrid cross earlier on, okay, the monohybrid cross had that 3-to-1 ratio. For a dihybrid heterozygote cross, we will always see the 9-to-3-to-3-to-1 ratio in our phenotypic ratios. Okay, this will tell us if we did all of this other stuff correctly. Okay, and again, this is just an example for you to go off of so that you have something to reference back when we do all of our problems in class. Okay, the problems in class are not going to be as easy as cross this one with this one. I'm going to give you a whole problem, and you're going to have to be able to pick out What are the heterozygotes? Are they homozygous dominant? Is it a monohybrid? Is it a dihybrid? Okay, so you've got a lot more things that you're going to have to be able to pick out. All right, now, there is another way to do a dihybrid cross. Okay, this is going to help us work into our big project that we're going to do for genetics, okay, which you'll get introduced to later, all right? But you're going to have to do a cross with more than 2 genes, okay? And more than 2 genes would be meaning that you're going to have a lot more than 16 boxes, and then you'd have to hunt and peck to find all of them, okay? So if you thought this was overwhelming with just the 2, okay, this is a little bit easier way to do it, okay? So this is what we call the fork line method. The fork line method, you're going to do 2 monohybrid crosses between the 2 genes that are the same. Okay, I picked the ones that are going to give us the most options because the heterozygotes always have all of the options. So again, this could also save some time if you're not crossing 2 heterozygotes. So if I'm doing a monohybrid cross between my R's, I set it up just like any old monohybrid cross. For my Y's, I set it up the same. Okay, again, my funny little thing on my Y's so that I can tell the difference. Then I fill across, and I fill down to give all of my different combinations. And yes, okay, some of you are going to be able to just look at this and picture what my options are. But again, you have to show me work. I have to be able to see the work behind it so that just in case your answers are wrong, I can give you some points. Okay? So this is step 1. You do monohybrid crosses for both. Then step 2 is you count them up with whole numbers this time. Not fractions, not decimals, nothing. Just whole numbers. So I've got 1 big R, 2 big R, 1 little r, little r. Shocker, I have the same over here for my Y's. Okay? So I have 2 big Y, or I have 1 big Y, 2 big Y, 1 little y, little y. Okay, so step 1, monohybrid cross. Step 2, count them up. Step 3 is the actual forking. Now this one you're going to want to give yourself some space, okay, so that you can actually fork everything off of it. So you're going to spread out your options. So if this had happened to be, say, 2 big R and 2 big R little r, I wouldn't have a third option down here. Okay, but because we do, we're going to write all of them out. Okay, then we get to the forking part. I'm going to fork my options for the Y off of each one. Okay, so here it is. I forked my 3 Y options off of my big R. Now I'm going to fork my exact same Y options off of my big R, little r. And then finally I'm going to do the exact same to my little r, little r. And yes, see, I'm kind of running out of room. So make sure on your pages, when you're showing me all of your work, that you're giving yourself enough space. Okay? Now it's like a multiple choice question. Okay? So you got to kind of picture that there are little times signs all through here. Okay? So it's 1 times 1, bring your letters with. 1 times 1 is equal to 1, and then I just bring my letters with me. So I got big R, big Y, big Y. 1 times 2 is 2, bring my letters with. Big R, big Y, little y. Bring my letters with. 1 times 1 is 1, big R, little y, little y. Doing the same down here. 2 times 1 is 2, big R, big Y. 2 times 2 is 4, big R, big Y, little y. 2 times 1, 2, big R, little y, little y. Down my last 3, 1 times 1, 1, little r, big Y, 2 times 1, or 1 times 2 is 2, little r, big Y, little y. 1 times 1 is 1, little r, big Y,
big Y. Okay? Best part about this one, okay, here's your genotypic ratio right here. You're already done. Alright, and yes, that says geno. Alright, I count them up. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Put it out of 16. It's like they're fractions. I'm done with my genotypic ratio. Okay? Then my last part would be doing the phenotypic ratio. Okay? And if you can look back, these look very similar to what we just did. Okay? Except now we're talking, let's say, round and yellow instead of tall and purple. Okay? So now we're going to be looking for anything that is round and yellow. Okay, and in pea plants, if you're not round and yellow, you're round and green. Okay, or you are wrinkled and yellow. I know, writing is horrible on this. Okay, first one, everybody's just going to have to work with me. Okay, wrinkled again and green. Okay, then we just count everything up. So again, round and yellow, both are dominant, so I'm going to count dominant, 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 no, dominant, 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 dominant. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so we should be adding up to 9 because again, it's a heterozygote with a heterozygote, so I should have my 9 to 3 to 3 to 1. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So I have 9/16, because remember my 16 is down here, that are round and yellow. Okay, then my next one, round and green, and round and green. 1, 2, 3. 3/16, leaving me just those to get from to be wrinkled and yellow. Again, 3/16, because I have my homozygous recessive, big, okay? So those are all going to be wrinkled because they have the homozygous recessive there, and then the one dominant allele. And finally, the 1/16 that leaves both of the recessives, okay? Homozygous recessive for wrinkled and for green. Alright? So those are two different ways to do your dihybrid crosses. Okay, so you can do the 16 boxes, or you can do fork line. Now this one you're going to have to do, unless you really want to do the huge boxes, when we have something that maybe could look like this. Okay, so there's your round, then you're also going to look for yellow, and then I'm also going to see what is my probability of having tall. Okay, so if you're doing a cross with 3 different genes, alright, you're really going to want to do fork line because it's going to take you a lot less time. Alright, so if I was doing this, then I would just be forking off my T's over on these. Okay, whatever they did, because I would have to have a whole nother box for my T's. Alright, and we will come back to doing this with 3 different genes, but I want you to be kind of thinking ahead and seeing which one that you like the best so that you can do our problems efficiently. Okay, next time we meet, we're going to talk about the other types of inheritance patterns. Okay, so that's going to be your incomplete, your codominant, your sex-linked, and these are the ones that kind of get to be a little bit more fun, but we've got to have the dihybrid and the monohybrid crosses mastered before we move on to incomplete, codominant, and getting into all of these other options that we have with the way that our inheritance patterns. Okay, your next question is going to be, when do we get to talk about humans? When do we get to talk about humans? We will talk about humans, I promise, but we've got to get all of this dihybrid and monohybrid stuff understood first before we move into more difficult concepts. Okay, alright, so I hope that you enjoyed this. I hope that this worked, and we will review all of the different problems when I see you in class on Wednesday. Thank you so much.